Now, why do proteins fold? Well, among proteins, structure is directly related to function. In other words, the three-dimensional shape that a protein takes on is directly related to the job it can do. This is especially important for proteins that have what's known as an active site. Here we have a three-dimensional model of a protein, and you can see that it kind of has this little cave in the side. It has a dent. That dent is known as the active site. Into this dent, a molecule known as a substrate will fit, and it will fit like a lock and key. And that's going to allow the protein to do something to that substrate, to break it or build it or move it or do something. If the shape of the active site is affected by a mutation or by damage, the substrate can't fit in there. The active site is ineffective, and the protein can't do its job. So among proteins, structure directly relates to function. Here we have 4 different proteins, each with different structures and different functions. The first is insulin. Now, insulin is a messenger. It is a hormone molecule. It's released by the pancreas when your blood sugar levels begin to rise. And so it will go out into the blood and it will travel around until it reaches the liver. It will bind with receptors on the liver cells and it will tell the liver to remove some of the sugar from your blood. The shape of this insulin is really important because if the shape isn't correct, it can't bind with the receptors on the liver cells, so it can't send its message. Here we have fibrin. Fibrin is used during times of injury, when you cut yourself, for instance. It's shaped like long strings, and what this will do is it will form an internal net across a cut. And if you look closely, you can see that there are red blood cells trapped in this net. So it kind of stops the bleeding at a cut until the coagulants and other clotting factors can kick in and make a proper blood clot. Here we have hemoglobin. Hemoglobin carries oxygen in your blood. To do this, it has to bind to iron molecules, and in order to do that, it has to have this particular shape. Again, it has 4 subunits, all of which hook together. Here again we have collagen. I mentioned it before. Collagen is shaped like a bundle of springs, and it's found in the deeper layers of your skin. It's what allows your skin to remain springy, to stretch, and to pop back into place. So 4 different proteins, 4 different structures, 4 different jobs. These things we call mutations can affect how a protein functions. Sickle cell anemia is an excellent example of a mutation that affects a protein. So to understand this, we have to understand hemoglobin. Here's the shape of a normal hemoglobin with its 4 subunits put together. Hemoglobin exists inside red blood cells, and because hemoglobin has this particular shape, red blood cells have this kind of a disc shape. Because hemoglobin is a protein, it's made of a string of amino acids, and they have to go in a certain sequence in order to fold up properly. In people with sickle cell anemia, a single letter on the DNA is changed. That results in a single amino acid being put in the sequence incorrectly. Look what that does to the shape. When this amino acid sequence folds up, it folds into this kind of weird shape. And inside the red blood cell, it sickles it. It pulls on that red blood cell and makes it kind of a crescent moon shape. Now, sickled cells like this don't carry oxygen as efficiently as the regular red blood cells. They also tend to kind of pile up and get stuck in the capillaries, so they can cause a lot of swelling and a lot of pain, especially in the joints. So this is an example of how a change in a single amino acid can have a big physical effect on the protein and on the cells in which that protein is found. Now, if proteins can be folded into a 3-dimensional shape, they can be unfolded. We refer to this action as denaturing. Denaturing occurs because of an exposure to heat or strong acids or bases. And exposure to these things breaks down the bonds that hold together the tertiary and secondary structures. So that protein is broken back down into its ribbon-like shape. Now, one of the major bonds that's broken during denaturing is a disulfide bond. That occurs between 2 sulfur atoms in the R groups of the amino acids. The effect of this is that sulfur is often released from a denaturing protein. So sometimes when proteins are denaturing, they smell a little like boiled eggs. In fact, that's exactly what happens to eggs as you boil them. You may have noticed after you boil eggs that there's kind of a ring around the yolk. Sometimes it's kind of a greeny looking ring around in this area. Well, that is sulfur that is building up inside that egg from the denaturing of the proteins. And because there's a shell on the egg, it gets kind of trapped around the yolk. It doesn't hurt you to eat that at all. It just looks kind of weird. Another place you might find denaturing is with hair permanents. If you've ever permed your hair and noticed that it smells like sulfur, well, that's because you're actually breaking down the proteins in your hair and reforming them into a different shape. And so sulfur is being released from your hair as a product of that. Denaturing is also a problem in cases of high sustained fever. If you get sick and you have a really high fever for a long time, the proteins in your blood and in your brain can begin to denature. That can cause organ failure. That's the reason that doctors will often try to bring a fever down by dunking someone in an ice bath or putting ice packs on the body, is to kind of break that cycle and prevent that denaturing. So denaturing is a powerful tool. It's also something you have to watch out for when you're sick. Hooray, you guys, you're finished! Arnold would be proud of you. You are now ready to attend class and put into practice all these great protein skills you've built up through this lecture. We'll see you then.